OsteoBites is a weekly osteosarcoma webinar and podcast presented by MIB Agents. This week, we are talking with Dr. Andrew Livingston, co-director of the Adolescent and Young Adult Program at MD Anderson Cancer Center and assistant professor, Department of Medical Oncology, University of Texas, MD Anderson. Our panelist is Florencia Pistrito of Osteosarcoma and Ewing Support Group. I'm your host, Anne Graham. Welcome to OsteoBites, everyone. Hope you have your snacks ready to crunch on because we have a really exciting session today. My exciting snack is vanilla double stuffed Oreos. My name is Anne Graham, and I'm an osteosarcoma survivor. I started MIB Agents in 2012 with the mission to make it better for kids with osteosarcoma. That mission continues today by providing direct patient support to patients and families, and by bringing together the physician, researcher, and patient community to work together, and by funding meaningful osteosarcoma-specific research. Awesome day at HQ today to have Dr. Livingston with us from MD Anderson to talk about his MIB-funded Outsmarting Osteosarcoma research. And also, as usual, we have Florencia Pistrito, who's our patient advocate. Amanda is not able to be with us today, so we can get started by Dr. Livingston. If you could introduce yourself, please.
Great, thanks, Anne. Hi, everyone. My name's Andy Livingston. Very happy to be here today to share with you guys a little bit about our research in osteosarcoma. So I am an osteosarcoma physician. I'm a medical oncologist, so I take care of teens, young adults, and older adults with bone sarcomas. And when I'm not in the clinic, I spend my time in the research lab working on new treatments for patients with osteosarcoma. So excited to be here and to share some of our work with you.
Great, thank you. Florencia?
My name is Florencia. I am the administrator of the group on Facebook for osteosarcoma and Ewing sarcomas patients, families, and carers. I am an advocate as well.
Thank you, Florencia. Dr. Livingston, you ready to go?
Let's do it. Okay. So I thought a lot about what could I tell you about in a brief time, a quick talk here today, and really thought this would be a great opportunity to share our group's work looking at the immune profiling, looking at the immune system, its interplay with osteosarcoma, and was very fortunate to present two talks at the recent Factor conference. So for today's talk, I basically slammed those two together to talk about what have we been doing in osteosarcoma to understand its interplay with the immune system, and how are we going to bring that to the clinic to help osteosarcoma patients. So I think originally my talk was titled or listed as CD73, the next immunotherapy targeted osteosarcoma. So we'll talk a little bit about that, combined with another talk I gave at the Factor conference, defining the immune atlas in osteosarcoma, where I was very fortunate to be selected for the Outsmarting Osteosarcoma grant. And really the goal is to think about how can we bring immunotherapy to the clinic for patients with osteosarcoma. So I think, you know, there's a lot of news, buzz, and excitement around immunotherapy and cancer. It's kind of everywhere you look. Jim Allison, who is a senior scientist here at MD Anderson, was a recent Nobel laureate for his work in this area. I think most people are familiar, but as a quick overview, what is the idea of immunotherapy? You know, basically what we're talking about is, can we use the body's immune system to attack cancer cells? And in some ways, cancer is very different than our normal cells, but in some ways it's very like those normal cells. And so early on, the immune system can recognize cancer as a foreign invader. It sends over these T cells to attack the tumor. But what happens is the T cells upregulate these molecules that are called immune checkpoints, basically the brakes on the T cells to say, whoa, 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 that tumor's kind of like us, we don't want to attack. And these are things called PD-1 and CTLA-4. So there's a new type of therapy called immune checkpoint inhibitors, drugs that target this, kind of shown in the middle of the slide here, called anti-PD-1, anti-CTLA-4, have been the initial types of treatments where they basically block these signals, release the brakes, and allow the immune system, specifically the T cells, to attack a tumor. And this has really been a promising avenue for treatment of many types of cancers. But of course, we've all been interested, can we take that same approach and use it for patients with osteosarcoma, for patients where the tumors come back despite normal treatment with chemotherapy and surgery? And there's actually been several clinical trials that have tried these strategies with various different drugs. And kind of summarized here, but really the punchline is that this approach really just hasn't worked very well for patients with osteosarcoma, with drugs called pembrolizumab, nivolumab, ipilimumab, a specific study of atezolizumab and kids and young adults with osteosarcoma. Really, we haven't seen that just using these type of immunotherapy drugs by themselves has just not been very effective. And so one of the big questions we had is, why is this not working? And of course, how can we make it better? So very quickly, very broadly, there's two main reasons that immunotherapy doesn't, or two kind of categories of why immunotherapy and immune checkpoint inhibitors specifically may not be effective. One is what we call intrinsic mechanisms. That are, what are the things going on within the tumor itself, within the cancer, within its DNA that may make it resistant to this approach. And the second group is kind of called extrinsic mechanisms. What's going on around the tumor? What's going on in that tumor environment? What are the other immune cells in that area that may be shutting down these signals, like immunosuppressive cells, or these checkpoint checkpoint molecules are upregulated? And so we fortunately did a big study looking at this same idea in about 50 patients with metastatic osteosarcomas, and we were able to do some really in-depth, cool profiling work and have our patient paper recently published. So if you want to really nerd out on the science, I'd encourage you to take a look at this study that we published, but I'll walk you through some of the major findings. When we looked at all of these tumors using RNA sequencing, what we were able to find is that, some osteosarcomas really are immunogenic. Around that tumor, within that tumor, there's a ton of immune cells there that are trying to attack the tumor, but for some reason that's not working. And we found out we could categorize the osteosarcomas into kind of three main groups. Those with a very low amount of immune cells, that's this blue cluster called C1, intermediate C2, or high C3, where we have all kinds of immune cells within those tumors. And basically what this is showing is that, different osteosarcomas have different levels of immune infiltrate and immune response. So the next question we ask is so what's really different between these osteosarcomas that have a lot of immune cells and those that really don't have many immune cells, and what some people will call hot or cold tumors. And in this plot, the hot tumors are these in the red, the cold in the blue, and we could ask this question, what's different? And we looked at these different pathways, and kind of not surprisingly, in the immune hot tumors, we saw very high expression of these immune checkpoints, things like PD-1, CTLA-4, and some other signaling pathways here. And then we could also look at what are some of the very specific genes that are either on or off that are very different between hot and cold osteosarcomas. So in addition to seeing that this PD-L1 thing is high, also called CD274, we saw some other really important genes that are interesting and maybe help us when we think about how to bring this to the clinic. In particular, one example is a gene called TGF-beta, which you can see on the top of the list here. And basically, what does TGF-beta do? Well, many things, but one thing it does is it can block the immune cells, these CD8 cells, from getting to the tumor. So even though those T cells are in the tumor, pathways like TGF-beta can prevent the immune cells from getting in there and doing what it wants to do, which is to kill the tumor. So that was really the basis of what we've done so far and led to our next project. And this is the project that was recently funded by the Outsmarting Osteosarcoma grant, where we're going to use a really cool new technique called single-cell RNA sequencing and TIL profiling to look at the microenvironment. What's going on around these tumors, particularly in the lung? So what is single-cell sequencing? Well, when you look at a lot of what's being done with next-generation sequencing and the data we already did, what I showed you is something called bulk RNA sequencing. And when you do that, it's basically like you throw the tumor into the blender. It's all there. You can figure out some of the things like I showed you. It's kind of like drinking your smoothie here. You say, yeah, I think I taste some blueberries, maybe some strawberries, but you can't quite figure out what's going on. That's really bulk sequencing. So what we want to do is use a technique called single-cell RNA sequencing to really basically pick this apart before it ever goes in the blender and figure out what's really in there. which of those cells are the tumor cells, which of those are those good T cells that are attacking the tumor called TILs, TILs, what's going on in the lung or the so-called microenvironment, these type of things. Can we really parse that out and look on a cell-by-cell level to understand what's going on in terms of the immune response to osteosarcoma? And one of the really cool things that this allows us to do is not just figure out what cells are there, but actually we can figure out and quantify them to understand which are these different cell populations, how do they interact, and this is an example from a different type of cancer. And why is this important? Well, it's kind of like being in a power play in hockey. Knowing which cells predominate, knowing which cells are there can help you to understand who has the advantage. We see good and bad cells there, pro-immune cells or immune suppressive cells. But when we look in this way, we can figure out, well, maybe it's those suppressive cells that are really tamping down the immune response. And that can give us some ideas about how to go after that in terms of cancer therapy. So in the first part of this study, what we're going to do is take patients who have osteosarcoma lung metastasis, tumor in the lung, who are already going to be going for surgery. So, very often we do surgery as a standard approach to getting rid of osteosarcoma lung metastasis. So patients who go for surgery, we're going to collect those tumors, take them to the lab, and we're going to do a lot of things with them, looking at sequencing them with what's called whole genome sequencing, the single-cell RNA sequencing I was just describing, and also look at what's going on the T cells. And the other thing we'll do is called immunofluorescence, which I'm showing here, which allows us to look at what are the different immune cells in that tumor and where are they? Are they around the tumor? Are they in the tumor? To try to understand where are the immune cells relative to the tumor cells. And the second thing we're going to do is to look at the immune cells in the tumor, what are called TILs or tumor infiltrating lymphocytes, and figure out what is it about those cells that were able to identify the tumor, get in there and start attacking it. What's specific to those cells? And one of the cool things that's being done, in fact, in a clinical trial we're running currently here at MD Anderson, is a type of treatment called TIL therapy, where basically we can take a patient's tumor, take it to the lab, we can chop it up into bits, extract the immune cells, the T cells, make millions of copies of them or billions of copies of them in the lab, and then we can infuse them back into the patient for this idea and goal that those cells will target the tumor and attack the tumor. And part of what we want to do so that's in the trial, but in the specific research we're doing, we want to try to understand what's going on in those TILs, which ones are helpful and not, and how can we make them more effective. So that's kind of the second part of what we're doing in this research study, is taking those same fresh tumors that were taken out of the lung, extracting the lymphocytes, the TIL cells, and really characterizing them. What do they look like? Which ones of these are recognizing the tumor, and what are they recognizing? Okay. Hope you're still with me. One last part here is talking about how are we going to bring these things to the clinic, or what are we doing in the clinic now? So there's a lot of interest in immunotherapy and osteosarcoma, various things going on around the country. I'll touch base mainly on what we're doing here at MD Anderson. The first is a current trial we have open, which is using TIL therapy I was just describing for patients with osteosarcoma. The second study is a study that is just about to open, and I'm going to talk a little bit more about, which is a new way of using immune checkpoint inhibitors in osteosarcoma, using a drug that targets PD-L1, another one that targets another signal, CD73. And I think really the next steps beyond that we're already developing. Some of these are using a combination of immunotherapy, like immune checkpoint inhibitors with targeted therapies. Can we add something to TIL therapy? Are there other ways to use immune cells using approaches like what's called CAR-T, TCRs, or NK cells? So just to share with you a little bit about our current study we're opening that targets CD73 in osteosarcoma. So CD73, probably something most people haven't heard much about, scratching your head. So here's a little overview of the many roles it has. But basically, CD73 is involved in what's called adenosine metabolism. And it's thought that it can help tumors to evade the immune response. And basically that this is a molecule, this is an enzyme that catalyzes this rate-limiting step to produce adenosine outside the cell. And in that case, when CD73 is high, we think that it suppresses effector T cells, those good T cells, the ones that should be attacking the tumor, and allows that these immune suppressive cells called Tregs and MDSCs, that you get a lot of these suppressive cells. So of course, the goal or idea is, can we block that CD73 signal? And if we do, can we increase the good immune cells, these CD8 positive cells, and then really turn down or shut off those immune suppressive cells? And if we do that, can that make other types of immune therapy work better? And that's the idea. So here's a little flashback to the data I was already showing you. So what we were able to do is go back and look at the osteosarcoma tumors. We had already looked at these groups of hot and cold tumors, 1, 2, and 3. And we already know that PD-L1 expression is higher in those immune-infiltrated tumors in C3. And we also can find out, or we also found out, that CD73 is also upregulated in that group. So giving us more kind of support to this idea that if we target both of these pathways, that could be effective for patients with osteosarcoma. This is just a super basic schema that shows what's going on, how are we going to do the trial, what will be involved. And of course, happy to share more details with anyone who's interested. But basically, this will be a clinical trial for patients who have metastatic, recurrent or metastatic osteosarcoma, patients who've had standard treatment and the tumor has returned. And when patients go onto the study, what we want to do, or we're going to ask patients to do, is to have a biopsy of their tumor, some additional blood sampling to let us understand which of the immune groups do patients fall into. And then we'll treat them with these 2 immunotherapy drugs, one called durvalumab, one called oleclumab. And we'll continue that treatment. We will follow up, and after the initial treatment period, we'll get a second biopsy of the tumor and start doing scans. So that way we can figure out 2 things. Obviously, the scans will tell us how much is this treatment helping any one patient, and the biopsies will help us to understand how effective is this treatment for patients with osteosarcoma, and which patients are specifically the ones who are going to benefit from this type of treatment going forward. So of course, we're excited to bring this treatment to the clinic to help patients, and really think the way the study is designed will really help us learn an immense amount about how to make immunotherapy work better for osteosarcoma. Oops, here we go. So I know that was a whirlwind tour. Tried to wedge a lot of work into a quick snapshot, and I have a lot of people to thank. A lot of this work has been done here at MD Anderson with the genomics group, particularly Per Wu and Hannah Baird, who helped with a lot of the analysis that I showed. And support from bioinformatics and the medical oncology group, Pediatrics. You can see everyone here. And really none of this work would be, we wouldn't be able to do this work without the generous support from a number of foundations, including the Quad W Foundation. The clinical trial I shared is specifically funded by the Osteosarcoma Institute. And we're very fortunate to also receive funding from Triumph Over Kids Cancer, the Antron Foundation, and CPRIT. So I hope I have left us a few minutes to tackle some questions.
You probably hate this question, so apologies for it right up front. How far out are we thinking this trial might be?
So the main trial I was talking about there, the CD73 immune checkpoint trial, is actually fully submitted. It's being reviewed here at MD Anderson by our IRB, and we're actually expect to have the approval within the week. And my hope is that we're probably about 6 to 8 weeks from starting to enroll patients. So it's very close.
Wow. Oh my gosh, that's so exciting. I'm so glad I asked.
Yes.
Can you describe the impact that these nonprofits, including MIB Agents, has on the ability to further research and discovery in a small rare disease community like osteosarcoma?
Oh, I think it's a great question. I mean, I think it's hugely important and very impactful. You know, as you're very aware, I think many people are aware, the funding for rare cancers and pediatric cancers has been less than more common cancers in the past. And when we look at projects that are specifically focused on osteosarcoma, this type of support and funding has really been critical to the work we've already done, and is the reason the next project will get done. So, I shared that it was many small foundations that helped support our sequencing studies so far. And this project really was a concept that had come together, but wasn't going to be able to move forward without the support of MIB agents. So I'm just incredibly grateful. And it's really the catalyst to do this type of work. And it's an interesting phenomena where we see that a small amount of seed funding, or not to suggest this is a small amount, but once you get that seed funding, it really is something that can really amplify, and you can get other donors, other support, institutional buy-in from the pharmaceutical industry, just by having that little initial funding to really get projects like this off the ground.
Yeah, agreed. I did not realize before we started funding research, before I had osteosarcoma, how impactful $100,000 is to someone's research. And we've seen in the research that we funded that those projects have gone on to get greater grants and more funding and are moving right along toward trial, which is huge. And I have to say, for MIB agents, we couldn't do any of this where we're able to fund osteosarcoma specific research through our family funds. So if you have a child or a family member with osteosarcoma, you set up a family fund with us, and you can designate that fund for research or for our programs for direct patient support, or funding osteosarcoma specific research. And that's how we're able to annually fund meaningful research for osteosarcoma. And I think it's had an impact on the osteosarcoma landscape of what science is being conducted. So thanks for the explanation on that. It's really something that I don't think a lot of people understand. I know I didn't understand it until I did. So thanks for your explanation. Florencia, I think we have a couple of questions. Do you want to?
I had a member of the group that was asking me, because I saw that you mentioned something about when the tumor that is extracted, that sometimes it's a hot mass. So he was asking a bit about the difference.
Yeah, so I think these terms are kind of the buzzwords or lingo around cancer immunotherapy. And hot and cold is kind of the words we use to describe tumors that either have a lot of immune cells in there, or ones where there's not much going on in terms of the immune system, the T cells and the immune system attacking the tumor. And so typically we say tumors are hot if they have a lot of immune infiltrate, a lot of these different immune cells within the tumor. And one of the things I didn't show here is that actually this idea about tumors being hot or cold has been quantified, or has been looked at actually across all different types of cancers. And what we know is that tumors that are hot are the type of cancers that have really shown the most benefit from immunotherapy. So these are things like melanoma, kidney cancer, non-small cell lung cancer, for example. Those tumors tend to be more so-called hot tumors or immune infiltrated. And so we think that some of these osteosarcomas look more like those tumors. And if that's the case, those are the type of osteosarcomas that were much more likely to benefit from immunotherapy.
Here's a question from a young woman who's a metastatic osteosarcoma patient. And she has two questions for you. One is, on this immunotherapy that you're thinking of, and in immunotherapy in general, does the patient lose their hair?
So typically, no. Immunotherapies, I think there is a little misconception that immunotherapies are easier than chemotherapy or have less side effects. The truth is they just kind of have different side effects. But one that's important and different is that patients receiving immunotherapy typically would not lose their hair.
Cool. Okay. She also wants to know if...
But what are you saying about my haircut?
Well, it's totally working for you. If you're a teenage girl, yeah, it's not the ideal.
Probably not so much.
Probably not so much. So if you have lung metastases and the patient has gone through MIB-ACC or mifemertide, would she qualify for this trial? And this is, I mean, really, I suppose it would be somebody in that situation, right? Because if you went through first-line treatment and it succeeded, and you didn't have any metastatic disease, you wouldn't need it in immunotherapy.
Right, that's true. So let me answer kind of that in a relatively broad way, and anyone who wants to reach out to me directly, you know, I'm happy to share my contact information as well, and I can get more specific details to people who are interested. You know, these trials are, as you said, specifically designed for patients who've had standard therapies and then have metastasis or have their tumors come back. We're really not at the point where we want to add immunotherapies to the initial treatments with drugs like doxorubicin, cisplatin, and methotrexate. But yes, patients who've had other treatments are typically eligible for this type of treatment or this clinical trial. And I think one of several questions come up. Can this be done in patients who have metastasis that may be removed surgically? You know, for the two clinical trials I mentioned, the TIL study and the CD73 study, patients have to have areas that are considered measurable. So if someone's had all their metastasis removed with surgery and they have no evidence of the cancer that we can see, they wouldn't be eligible for this type of study, although there are other studies available. And I think that answers the question.
Yep, I think that does it. Florencia, any questions?
There is any trial going on in the US with MIB for people that have lung metastases?
I think many people will be familiar with that. MIB is not currently approved by the US FDA. And actually, partially because of that, there's not a mechanism, or right now, MIB is not being used in clinical trials in the United States. And so there are various mechanisms where patients, even within the US, have been able to get access to that medication. But right now, it's not approved by the FDA and not being used in any clinical trials within the United States that I'm aware of. I think there's been a lot of interest for a long time in this drug. And in fact, one of my mentors when I was first starting out is Jeannie Kleinerman, one of the amazing osteosarcoma researchers who helped pioneer this work with MIB. And so along the way, we have talked about, could there be strategies that combine that with these immune checkpoint inhibitors? But so far, that really hasn't come together as a possibility.
Okay, another question. It sounds like it's only for non-resectable tumors for the trial since they biopsy it twice, or can you undertake this trial with a resectable tumor?
So that's a great question. You know, all clinical trials have about 300 rules. It's a very lengthy list of eligibility criteria. But the short answer is actually yes, that patients with potentially resectable tumors could go on this study. But for most of these studies, they have to be willing to stay on the treatment for a minimum amount of time before going to surgery. Meaning, if the treatment's working, we would continue that treatment usually for 4 or 6 months before patients would be undergo a surgery to remove the tumor. Because part of the idea is if that tumor is in place, it will allow us to better understand how effective is the treatment. But each trial has different rules, and I'm happy to discuss specific patient, with each patient specifically if these would be options.
This is assuming fresh tumor versus frozen? Does it have to be fresh tumor?
So a couple, yes and no. So a few different questions. You know, for the research study where we're looking at the lung metastasis, that's going to require fresh tumor, but that study will be specifically, a laboratory translational study, and for patients who are already planning to have lung surgery as a standard of care, patients who they need to go for lung surgery because that is the right treatment of their disease, and we'll take those tumors to the lab. That requires fresh tumor. For a lot of the studies, like the clinical trials with the biopsies, oftentimes these studies will allow us to use archival tissue or FFPE tissue initially, and then get a new biopsy during the treatment. So oftentimes we can use previous tumor tissue from a surgery or a biopsy in the past to kind of avoid an initial biopsy. But again, the specifics are different for each study.
Right. Okay, I had just a thought occur to me last week. No, sorry, not last week. The week before, we had a OsteoBite session with Dr. Trucco and Dr. Loeb about upfront surgery for osteosarcoma. So not going through the 3 cycles, first having removal of the tumor. So you have this large as possible virgin tumor that hasn't been affected by chemotherapy. And then you have that tumor to study, not only for researchers everywhere, but also for that patient. It sounds like this would be helpful in this particular kind of study where you would have that virgin tumor, and you'd be able to compare and contrast with lung mets down the road or any other metastatic disease.
Yeah, I think it's a really good question, and it's something that's kind of a little bit controverted in osteosarcoma. There's a lot of advantages to the approach you're describing, and particularly, I think the question is what's more important? Is it understanding what's going on in that tumor in the beginning, before, like you said, before it's been exposed, before it's been hit with these standard chemotherapies, or is it more informative to understand what's left behind when the chemotherapy is done? And it's a question scientifically we don't know the answer to, but one theory, one idea is, if you give chemotherapy and you kill the majority of the tumor, it's probably not those cells that are dead that are going to cause the problem. It's those ones where you got the chemo, but they didn't die. And are those the cells that come up as metastasis? Actually, there haven't been enough well-powered studies where they take, new diagnosis, post-chemo tumors, and metastasis from the same patient to really understand what we call the clonal evolution of the tumor. Mm-hmm. So we don't really know the answer to that. So I think you get a ton of great information by collecting a tumor where you haven't had chemo, but it's not necessarily better or worse. It's just different. But you could learn a lot. You could definitely learn a lot from that approach.
Yeah, we have so much to learn in osteosarcoma. It seems
we
like
do.
if you're in that zone where this wouldn't cause any harm over the long term, I think it'd be so valuable. I would be for it, certainly in my own case, and I can't see why anybody else wouldn't be for it either, especially to have that kind of timeline, like a tumor timeline. This is what you look like when you came to us. This is what seemed to be working. This is what's not getting worked on in this process.
Yeah, I agree. I mean, I think we can learn so much by looking at what's called longitudinal sampling. And I think, to me, I still think there's a lot of value in giving that initial chemo and looking at what's that percent necrosis, how well did that chemo work. But even if you're using that approach, I still think that getting and collecting a fair amount of the tumor before you ever start treatment, even if that is kind of using a multiple biopsies and then getting the tumor after chemo, and then if it were to come back, I think all that information is so interesting and informative, not only to the science, but that, I'm hoping we'll get to an era where knowing that level of a detail in a specific patient can help us better understand what's the right treatment for that patient. So,
right.
I think that's a common theme when you ask osteosarcoma researchers, and everybody wants more tissue and more studies and more sequencing. And I think because we know there's a lot out there that we don't know.
Yeah, true story. true sad story. We have partnered with Childhood Cancer Project and CCTDI to fund legacy autopsies. So, and the interesting thing about this legacy autopsy program that has been informed with several legacy autopsies by young adults who have passed from osteosarcoma, is it's the kids who are driving it. It's the person who has this disease who's going, please use my body for good. Use whatever information you can get from what's left of me for osteosarcoma research. And for speaking for myself, having had osteosarcoma and just had that dark night of the soul where you're like, oh my gosh, if this doesn't work, there's nothing, what else do we do? I remember saying to my doctor, just use me. Like, if I'm going anyway, do something. We have to be able to do something, because I'm looking around me at these kids with osteosarcoma, and we have to do better for them. We have to do better for them.
I totally agree. I mean, I think what a powerful statement for a kid with osteosarcoma to say that they want, that they want to, be able to contribute in that way. And it's just really, it's a really amazing testament.
Truly. The kids that I was treated with, I was treated as an adult at 43 in the Pediatric Cancer Center. And the kids I was treated with I told my husband one day, it was like climbing the top of the mountain, expecting to find a wise old man, and I found a child. They're so brilliant and such giving of themselves to try to. They're always in their own illness. I'm sure you see this every day in pediatric cancer, looking around and going, "Who can I help today?" Even though they're suffering greatly themselves, we do end of life missions. Many times when we call a child and we say, okay, what can we do for you? We do these experiences or items of comfort or entertainment when a child enters hospice. And 9 out of 10 times that child will say to us, I don't need anything, but there's this kid I was treated with who they don't have anything. Can you see to this child's needs? Or can you fund research? Or it's always about somebody else. Like, I shouldn't say always, 9 out of 10 times it's about somebody else. How do I serve somebody else in that moment when they're in hospice? Anyway, the goal is that we have no kids in hospice anymore, and this becomes a curable, treatable disease that goes the way of the dinosaurs. That's the goal.
That's the goal. I agree 100%.
Yeah. So thank you for your incredible work that we're so proud to fund and be behind you on in every possible way. We're so excited about it. So thank you for working so hard and going really above and beyond your time in the clinic to undertake this research to make it better for kids with osteosarcoma. Really extraordinary. Thank you.
Certainly. Well, I really look forward, once we have some more findings to share, once we have the results of that work, I really look forward to coming back and sharing with all of you about, what did we find and where does that take us next?
Well, we've got to come visit you soon too.
Come on down. Well, okay.
Yeah, I know. Not coming now.
Come on down.
Okay, so thank you again. And I want to invite everybody next week. We have Dr. Matteo Trucco of Cleveland Clinic. He'll be here talking about his MIB Agents Outsmarting Osteosarcoma Award. It was for disulfiram to overcome chemo resistance in sarcomas. So that should be exciting. Be sure and sign up for that. And also wanted to let you know that this OsteoBite session and all OsteoBite sessions are available on podcast and on our YouTube channel. You just have to go to our website, hit the blue bar on the top of the website. It says OsteoBites, and you'll be able to get all of the information there. And also really excited to tell you that we have funding from the Zimmerman Foundation for our book, Osteosarcoma from Our Family to Yours. It was written by MIB Agents Programs Chief Liz Vallejo. Our contributors are Dr. John Healy, orthopedic oncologist at Sloan Kettering, Dr. Matteo Trucco from Cleveland Clinic, Dr. Alana Church from Dana-Farber and Harvard Medical School, Maureen Smart, who's our licensed clinical social worker on our team, Jim Riley, patient family, and me. It's available for free download on our website, $5 shipping and handling if you want just one copy. If you're an institution and you want lots of copies and you're going to use them and pass them out, then we just ask that you cover our shipping, and those can come to you. So again, thank you for joining us today. And of course, Dr. Livingston and Florencia, thank you for what you do every day for osteosarcoma patients and families. And thank you for attending, and hope to see you next week. Stay safe, everyone. If MIB agents can be of help to you, please let us know. Together, we make it better for osteosarcoma kids everywhere. Join us next week when Dr. Matteo Trucco of Cleveland Clinic joins us to talk about his MIB Outsmarting Osteosarcoma Funded Research, disulfiram for metastatic sarcoma. Until then, make it better for kids with osteosarcoma. Visit our website at mibagents.org.